Good afternoon, everyone. I'm Kelly Reynolds, and I have been working on this project really with Hiroki Ando and Louisa Eichner, but this is Hiroki's baby, so I think Hiroki's in the audience somewhere. There he is. Yeah, so this is a project passion for Hiroki that he's put together, and we received some One Health funding to really advance this initiative. So I want to share with you some of the background for this project. So zoonotic infectious diseases have historically caused pandemics, and recently their threat has been growing due to increased interaction between humans and animals. So influenza A is one of the zoonotic infectious diseases infecting humans, but it also infects pigs, birds, and other mammals. And influenza virus causes significant impact around the world, as you can see some of the data on this slide. Certainly in the USA as well, and the virus is included in the list of one of the top 10 threats to human health in a WHO report. So the reason why influenza viruses cause endemics every year is the high frequency of evolution. So you have to get your vaccination every year because there's mutated strains and different strains that circulate that you maybe don't have immunity to. So that's one of the reasons why. Oh, it's not in presentation. Okay, are we there? Yeah, there we go. Okay, so that's one of the reasons why influenza A is such a concern because of its rapid mutation. So the One Health approach aims to balance and optimize the health of people, animals, and the environment. And by applying this type of approach to surveying influenza A viruses, we can get useful epidemiological information such as the spatiotemporal pattern of viral circulation in the environment and how these viruses are interacting between these different environments, people, and animals. So this information is effective for public health authorities to, one, prepare at the beginning of a pandemic or an endemic, and also to identify subtypes of viruses that are likely to be dominant for a particular season or emerging in future pandemics or endemics. So determining which pathway should be shut down to prevent emergence of newly pathogenic viruses is another approach, another goal of why we would apply this type of One Health approach to influenza A virus monitoring. So to obtain this information, we really need more comprehensive and reliable surveillance, epidemiological surveillance. And generally, these surveillance efforts are classified into two types, either individual testing surveillance or environmental surveillance. So one of our goals is to really kind of merge these two technologies so that we can get better, more advanced predictions of what might be happening in the population so we can implement public health controls sooner. Okay, so environmental surveillance has recently gained a lot of attention after the successful application of wastewater-based epidemiology in the SARS-CoV-2 pandemic. And wastewater-based epidemiology is an effective tool for this environmental surveillance goal that we have to really understand the prevalence of an infectious disease in a human community. We can analyze wastewater samples that contain a virus that's been shed from infected individuals, and studies have clearly correlated SARS-CoV-2 concentrations in wastewater with clinically confirmed cases. So similar demonstration studies have been reported around the world, and the application of wastewater-based epidemiology, or WBE, has been expanded to other infectious diseases, including influenza viruses. Regarding environmental surveillance for influenza A viruses, previous studies have reported the successful quantification of IAV concentrations in wastewater and strong correlation with clinically confirmed cases. And successful detection of IAV from environmental water are collected from wetlands and ponds where birds are migrating every year has also been used in previous studies. So the environmental surveillance for IAV is expected to be possible, and also by performing this type of surveillance together with WBE and environmental water surveillance, we expect to gain some insights around spatiotemporal patterns of influenza A virus in the environment and the evolution of these viruses in both humans and animal populations, and to also understand the environmental factors that are associated with influenza A virus prevalence in the community. So this information is really important for preparing for the next potential pandemic, especially involving IAV viruses. And in addition, the environmental surveillance system can be applied to other zoonotic infectious diseases. So, let me put the stuck here. Okay, so getting to our research objective around this project, we aim to develop environmental surveillance systems to track IAV circulation in the environment, looking at subtypes during endemic seasons, and to elucidate the interactions between animals, humans, and the environment. So right now, the lack of methods developed for doing this that are optimal for detecting IAV in different types of water sources is what we're working on. So improving the methodology, and there's really not a lot of studies looking at the temporal or seasonal impacts of IAV concentrations in environmental waters and what the dominant IAV genotypes are in the environment. And so there's also a lack of studies looking at the detection of IAV in wastewater, especially if you're looking at swine or poultry farms and what those different strains might look like in those types of populations. So what we have available to us is a data set. My thing's saying, it's saying restart, updates are required. I think I can just bypass that. It looks like it's blocking my slide. Okay, so we'll wait for IT for a moment, but I'll keep talking about this. So right now we do have a data set available to us monitoring 10 plus states looking at influenza A virus phylogenetic trees. And there has been groups that have taken the wastewater-based epidemiology data from the 10 states in the US and really characterized what these strains look like. So that was part of our goal originally with IAV, and another group has already published some of that information. So one of the things we expect to do in our study, it won't let me advance because of this update, I think. Maybe I can say restart in 5 minutes. See if that keeps us on track. Okay, yeah, it's not letting me advance, but so because some of this work with IAV has been done, like there's a lot of people working on it and kind of advancing this area of technology, we intend with some of our funding from the One Health grant to expand our surveillance to West Nile virus. That's a virus that's really impactful in our region in particular, and it's another way. It's another model organism that we can continue to develop the methodology around. So looking at the overview of our research plan, the first aim of our study is to develop an environmental surveillance system for IAV, and this phase is expected to take about 1 to 2 years ultimately. So in this step, we'll be conducting laboratory experiments to validate and establish a detection method for IAV in environmental waters. And using this established method, we'll collect water samples around Arizona and possibly California, depending on kind of how well things are advancing. And we're going to be looking for those dominant IAV genotypes in these water samples. So by comparing results from the environment that we find in terms of different genotypes with clinical surveillance testing, we can start to see what the relationship is between those two different environments for tracking and predicting future outbreaks. So our second aim is to elucidate how IAV circulations and interactions are happening between animals and humans. And in this phase, we'll expand our environmental surveillance to potentially other states in the US. So we'll need to write some other grants to kind of get this funded. But eventually, we'll have this long-term data set that can help verify our system for elucidating meaningful epidemiological information on a broader scale. Okay, so the first challenge in this approach is the selection or development of a highly sensitive viral detection method for IAV or any other virus in wastewater and environmental samples. And our research group has recently developed a highly sensitive detection method called the direct absorption extraction method, or DAE. And this method consists of concentrating using electronegative membranes. We concentrate the RNA from the samples that we're targeting. We do an RNA extraction directly from that membrane, and then we do an RT preamp procedure followed by quantitative PCR. And what we're doing is testing this in spiked samples, and in these spiked experiments, we confirmed that the methods we've developed are 100 times more sensitive than previous methods, which include ultrafiltration and PEG precipitation. So our method is much more sensitive for detecting respiratory viruses in wastewater, so we're really excited about applying this method more broadly and with different types of water samples. And in addition, we've confirmed that our method enables the detection of SARS-CoV-2 RNA from wastewater at about a 50% probability when clinical confirmed cases exceed 0.69 per 100,000 population. So if we have less than one case per 100,000 population, we can detect the presence of those viruses in the wastewater with a 50% probability. So this is orders of magnitude lower than what previous studies have been able to show. So in addition, using the DAE method, we've already succeeded in tracing concentrations of infectious disease viruses, including SARS-CoV-2 and influenza A viruses, over long periods of time. So this data has already been published. These are some of the references here from Hiroki's group when he was in Japan, and we're building on those methodologies continuously. So the DAE method has been shown that we can use it for determining IAV concentrations and identifying these dominant influenza A subtypes in wastewaters, and we aim to continue optimizing its use and expand its application as we've proposed in this. Well, I'm getting the message again. Let's see if we can pause the restart request. Okay. Okay, so although we've confirmed the applicability of this DAE method in wastewater, we have not confirmed the use of this method in other environmental water samples. So that's one of our first steps that we're investigating now. The piece of equipment that we really needed to achieve our sensitivity results from the previously developed methods just came in last week. We had ordered it some time ago, and so we're really ready to ramp up with our environmental sampling. We've been setting up some of our profiles and templates for the epidemiological tracking of and predicting of potential outbreaks, mapping that with the clinical cases with the databases that have been available, and now we're ready to start validating some of our results with more improved methods with environmental sampling. So different environmental waters are different than wastewater samples, and so everything has to be re-optimized in these different types of samples. So although we have not yet tried to detect viruses in different environmental waters, we have succeeded in quantifying these concentrations of SARS-CoV-2 in treated wastewater, so different types of wastewater, which has lower amounts of solids, and it would be more similar to some of the environmental waters that we're going to be targeting. So we're anticipating high success in the development of these methods. But after we achieve that and testing some of those methodologies, then we will investigate also the survivability of influenza A viruses in environmental waters and also on our filter membranes so that we can inform our utility of using this methodology for transporting and banking samples for future analysis in other states. So eventually we'd like to expand this beyond Arizona and California and move into other states. Okay, so this is just an overview of what we hope to accomplish after we've done more of this methods development in our laboratory experiments. We expect to conduct environmental surveillance outside of Arizona, moving into California and potentially these other states, as I mentioned. And the testing surveillance, we will collect environmental water and wastewater with the sampling method we've shown that you can see in this slide. The collected samples will be analyzed with the validated method, and this pilot data will be evaluated again for the temporal patterns of IAV concentration, and we'll also be looking more at genetic subtypes. Okay, so this is just more of an example of some of the states that we hope to target more long-term. Part of what we'd like to do, and I mentioned our goal originally was to develop this evolutionary tree of the different strains of IAV, but since some of that work has been done and published, it was from a group out of Ireland. So there's more that we could do towards that, but we also think something that would be more novel for us that we want to add in is, again, targeting West Nile virus. So look for that to be a bit of a modification where we'll start collecting some pilot data around West Nile virus in our particular region. Okay, so I think I'm near the end of my slides here, but this is just more of a schematic looking at the environmental sampling plan that we'll be coordinating, in particular with the bird migration pathways in North America and looking at states with the highest avian IAV cases. So you can see on the left side, you have in the colored map of the U.S., you have these zones of where the birds are migrating. And then on the right side, you can see where the most prevalent avian influenza A virus cases have been documented in the U.S. And with the red circles, those are kind of the more extreme regions. So those would be states we would like to start targeting initially as we expand. And so what we hope to achieve out of this is, these are just a list of some of the anticipated research papers. Some of them are focused on methods development and improvement. Others are focused on surveillance data, the pilot data, looking at a small number of states, and then identifying additional environmental factors that may be different across different states as well in the U.S., but just demonstrating that this environmental surveillance and this One Health approach, looking at the environmental waters where the animals are, and also looking at different environments for different types of animal populations, and really tracking this phylogenetic tree where we can see where these patterns are, I think will be really helpful. So, and then again, applying this to other zoonotic viruses. So that's our approach. That's our goal for this project. There's quite a bit to do in terms of collecting that pilot data, developing the methods, and then we're really excited to apply it. So we're thankful to the One Health group for helping to fund this initial work, and we think we can be very successful in achieving larger grants and broader funding to achieve these goals. So thank you.